Here is a short video demonstrating the process of creating a transgenic sheep which carries a gene for extra wool production. DNA is extracted from the sheep to undergo genetic modification. The DNA is cut open using restriction enzymes. The insulin-like growth factor 1, which is an ovine gene, and the mouse high-sulfur keratin promoter are incorporated into the sheep's DNA using DNA ligase. The modified DNA is inserted back into the nucleus of the sheep cell. This newly genetically modified sheep cell is propagated for further use. Egg cells from a sheep are removed. The egg cells are transformed. The nucleus of the cell is removed, but the cytoplasm, mitochondria, ribosomes, and various cellular organelles remain. This will be important later on. The nucleus is discarded as it is of no use. The transgenic sheep cell is then modified. Like the egg cell, the nucleus is removed from the cell. However, this time the nucleus is kept and the rest of the cell discarded. The nucleus from the transgenic sheep cell and the empty egg cell are then fused together to make a fertilized embryo. Genetically modified, it has a complete set of organelles, a nucleus, and the correct number of chromosomes. This diploid fertilized cell is ready to undergo mitosis as the embryo contains donor DNA. Multiple embryos are propagated so that more than one can be implanted into the surrogate mother. This is to ensure at least some of the eggs will successfully develop, as there is a high failure rate in the uptake of the eggs. Any lambs born from one of these embryos should express an excess of the insulin-like growth factor 1 gene, stimulated by the mice keratin promoter, and, as a result, should have a phenotype of increased wool production. In a study carried out where this exact experiment was conducted, the transgenic offspring resulted in an average fleece increase of 6.2%.